Hi, good day, dear colleagues. This is Dr. Jihad, and our today's lecture is a continuation to the series that we have started, What Surgeons Should Know. And today we are going to talk about physiology of the colon, functions of the colon, and only the colon. We are not going to talk about the functions of the small bowel or the function of the rectoanal region. We previously defined the colon as the ascending, transverse, descending, and sigmoid colon. And we will see that it has a few functions that is easy to review. Just a brief introduction to this lecture. When we want to talk about the function of the colon, this will indirectly lead us to talk about the innervation of the colon and the contraction of the colon, because it will help us in understanding some functions of the colon. Just briefly to discuss what function do we have that the colon participate in. Of them, of course, is absorption of water, of the main important function of the colon, that absorption of water and some electrolytes such as sodium, and exertion of potassium and bicarb. And this is actually maybe influenced by other mineralocorticosteroids such as aldosterone, as we will see. And you can imagine, to understand that the colon is a site for absorption of water, that about 2 liters of small bowel content enter the colon through the ileocecal valve. And of course, not all of these 2 liters will be passed with the stool. So there is absorption of water and electrolyte in the colon. We will see that it is mainly in the right colon. This is first. Also, the colon briefly participate in in the production of some nutrient material, of them the short chain fatty acid. We know butyrate, which is the main energy source for the colon. This is actually produced in the colon by the bacteria in the colon, the microbes in the colon. Also, some vitamins such as vitamin B are produced in the colon. Vitamin K also produced in the colon. And we know that babies and newborn who have no bacteria in their colon have deficiencies in vitamin K, as we will see in the next few minutes. Also, of the function of the colon is a passage of stool toward the rectoanal region, and storage of fecal material before evacuation, especially in the left colon. Okay, you can see here that storage of feces, absorption of vitamins that are produced by bacteria, electrolyte absorption, reabsorption of water, and you can see here that absorption mainly in the proximal half of the colon, while in the distal half of the colon they call it the storage colon, where they store the feces before it is excreted. Just a rapid review of the innervation of the colon. Actually, innervation of the colon, you can see that the nervous system, the nervous system is divided to central and peripheral. Central nervous system, actually this we mean the brain and the spinal cord, and we have many reflexes, as we will see, that control and participate in the motility of the colon: short arc, long arc reflexes, of them the gastrocolic reflexes. This is mediated by the central nervous system. The peripheral nervous system, which is divided to somatic and autonomic, actually somatic have no rule in the colon. If we are going more distal in the anus, we know that we have somatic innervation in the anus, in the skeletal muscles. But in the colon, it is the function of the autonomic nervous system, which is divided to sympathetic, parasympathetic, and enteric. And we know from the university years that sympathetic is inhibitory, parasympathetic is excitatory, and actually sympathetic is inhibitory to the colonic wall, and it's excitatory, enhanced contraction of the sphincter. Again, we are talking about the colon, not the anus, and because it's different physiology, different innervation, different entities. Just okay. Sympathetic, parasympathetic. We know fight and flight for sympathetic, rest and digest for parasympathetic. Enteric nervous system, we will see and we will discuss. We know that the enteric nervous system consists of Meissner and Auerbach plexuses that are present in the wall of the colon, and this will lead us indirectly to talk about the layer of the colon in this lecture, and how these cells or how these receptors will participate in the colonic motility. And remember that there is the interstitial cell of Sajjal that is considered the pacemaker of the GI motility. Okay, what I want to add also that we have two type of contraction in the colon. We will talk about them. These are the segmental contraction along short segments, and these are responsible for mixing and increasing the absorptive capacity of the colon, while the contraction responsible for moving the fecal continent or the colonic continent toward the anus for evacuation is called the propagated contraction, and can be classified to low amplitude, high amplitude propagating contraction. Okay, of course we are going to talk about the bacteria of the colon, as it is essential in the function of the colon. We say that bacteria participate in the production of vitamins, short chain fatty acid, etc. Okay, the feces amount is about 100 to 150 gram per day, and actually this is increased by high diet. Most of the feces, up to more than 50%, actually is of water, and the water content of the feces, or about 100 to 200 of water, is excreted with feces. And the other component of the feces, as you see, is bacteria, undigested fiber, fat, inorganic matter, protein. These make actually the 100 to 200 or 150 gram of feces that are excreted. We know that the brown color is due to the bilirubin. And okay, let's review. Okay, actually the pH of the colon, actually the content of the colon due to the bacteria is acidic, but with the exertion of the bicarb it will neutralize. We said that the colon will secrete the bicarb inside the colon, and this will neutralize the pH of the content of the colon. As you see that the colonic epithelium, which we will discuss, we will review it in the next few minutes, absorb water, sodium, chlor, short chain fatty acid, and secrete bicarb, which is responsible for neutralization of the acidic environment inside the colon. Also, it secretes potassium, chlor, and mucus, which will enhance the movement of the feces. Traditionally, or classically, the proximal colon is the site for storage, site for mixing, site for absorbing water and electrolyte, while the distal colon, it's like a conduit, work like a conduit for more, it's called the storage colon. Yes, the distal colon can participate in the absorptive mechanism under certain condition. Okay, you can imagine, this is a nice information, that the colon receive about 1.5 liters up to 2 liters of fluid from the ileum. So you can imagine that not all this amount is excreted or is evacuated. So there is absorption in the colon. All absorbed except only 100 milliliter of fluid, 1 to 200 milliliter of fluid, which is excreted with feces, and 1 milli equivalent of sodium and chlor are excreted, not absorbed actually, and excreted with feces. And actually this can increase and decrease if the patient have, for example, decreased in the small intestine absorptive capacity, so the colon can compensatory increase the absorption of sodium and chlor. Okay, let's move to the epithelium. Okay, the colonic wall anatomy, or let's say the histopathological anatomy, it consists of mucosa, submucosa, muscularis propria, and serosa. As you see here, this is the mucosa, this is the submucosa, this is the muscularis propria, and this is the subserosa and serosa. Okay, the mucosa itself consists of 3 parts. These are the epithelium, and we will talk about the epithelial cell because there is some differences in the epithelial cells in the colon from the other GI tract. We will review that. That will correlate with the function of the colon. Okay, so the mucosa, it has the epithelium, as you see here. This is the epithelium. A little bit out we have the lamina propria, and this layer is the muscularis mucosa. As you see here, this is a nice picture showing the layer of the colon. We have the mucosa, which consists of epithelium, lamina propria, muscularis mucosa. We have the submucosa, we have the muscularis propria, and serosa. Okay, adventitia, if we are talking about it's serosa actually. What I want just to alert you that we already talked about the enteric nervous system, and we said this is the Meissner plexus, Erbach plexus, and interstitial cell of Sajal, and you can see here that the Meissner plexus is through the submucosa. It is in the submucosal level. Okay, actually these have 2 different functions. We will review them. Erbach or mesenteric plexus, it is between the circular and the longitudinal muscle of the colon. And to explain for you, we have circular layer in the colon, which is all over the colon, sorry, and continues to the rectum and anus, and even in the anus it forms the circular muscle, it forms the internal anal sphincter, while the longitudinal muscle in the colon, it is the tinea coli. It is a condensation of muscle in three parts, and they are shorter than the colon itself, and because they are shorter than the colon itself, the haustration will appear. Okay, this is the longitudinal muscle. As we said, circular and longitudinal. Between them, we have the Erbach plexus. We will talk about the goblet cell, and we know that the epithelium of the colon is columnar epithelium up to the transition zone of the anus, where there is a transition from columnar epithelium to squamous epithelium, whatever keratinized or non-keratinized. Okay, what else we want to add regarding that? We talk about the submucosa and its three component. The mucosa, the submucosa, it's vascular. It's here we in the submucosa we find the vascular, the lymphatic, the Meissner nerve plexus, lymphs, vascular in the wall will be in the submucosal layer. These are loose connective tissue with collagen and elastin. We said that between the circular muscle, which is from the cecum up to the anus that form the internal anal sphincter, and the longitudinal muscle, which are the tinea coli, which actually ends in the rectum, actually we have the Erbach or the mentoric plexus, and throughout the muscularis propria we have the interstitial cell of Sajjal, which are responsible for gest tumor. Okay, let's move a little bit down and let's talk about the epithelial cell and how it differ and correlate, how it differ from other epithelial cell in the GI, and how it correlate with the function of the colon. Okay. We said that the most inner layer of the colonic wall is the submucosa, and it consists of the most inner layer is the epithelium, then a little bit outer the lamina propria, then the muscularis mucosa. So the epithelium and epithelial cell are in direct contact with the colon content, and actually we have only three cells or three type of epithelial cells in the colon. These are the enterocyte, goblet cell, and neuroendocrine cell, while in the other GI, such as in the small bowel, we have a variety of cells, but this will actually correspond to the function of the colon. As you see, we have three types of epithelial cell. Enterocyte, which is, we know that the epithelium of the GI system from stomodium, from the beginning of the esophagus up to the transition zone, is simple columnar epithelium, and enterocyte participate in absorption and secretion, not digestion. No digestive rule in the colon. Goblet cell, these produce mucus and facilitate passage of stool, and enterocyte and goblet cell make the majority of the cells in the GI, in the colon, up to 95%. Neuroendocrine cell or chemoreceptors, these are cells that actually initiate the local digestive reflexes, can detect harmful substances. For example, if we have feces in the colon that can harm the epithelium, it will initiate reflexes that will initiate the defecation mechanism. and it initiates protective response. Okay, the epithelium of the colon is renewed every 4 to 5 days, as you see, by dividing cell. As you see here, we have, this is the epithelium and the crypt. I have a picture, just I want to show to you. Okay, here. This is actually, they compare the small intestine and the large intestine. And one of the main differences that we have no villi in the epithelium of the colon. We have no villi. The epithelium consists of epithelial surface and crypts, no villi. So this is one of the main differences between large and small intestine, and actually villi are responsible for absorption and digestion. This is not required in the large intestine. To show you, to understand this, you can see this is the epithelium of the small bowel. Can you see? This is the villi. This is the projection above the epithelial surfaces called the villi, and this is the crypt. In the colon, we have only crypts. Okay. What else we want to add? That the crypt at the base, while the columnar, or the epithelial surface at the top, it's logically that the what is in the base is highly proliferative, undifferentiated. The crypt cell is highly proliferative, undifferentiated, while what is in the epithelial surface is well differentiated and low proliferative capacity. What else we want to add regarding this? Let's go back. Okay, these are the crypts. This is actually not villi but the epithelial surfaces. You can see that the colon, simple columnar, absence of villi, presence of goblet cell, deep intestinal gland, and the crypts. Okay, they talk about the mucus secretion, which is mainly by goblet cell in the crypts, clips of Leper-Koch, absence of villi, no digestive enzyme in the colon, presence of goblet cell that secrete mucus. Mucus can facilitate movement of feces and also provide adherent medium for holding the fecal material. Okay. We previously said that the colon absorbs sodium and water and secrete bicarb and potassium, and we said that this correlates with the function of the colon: absorption of water, which follow the sodium by the osmotic pressure, and bicarb is secreted, also correlate with the neutralization of the acidic environment in the colonic lumen due to the presence of bacteria. And this is actually under regulation of aldosterone, and you can imagine it like that. The stool is high in aldosterone, low in potassium relatively due to the aldosterone. Also, it absorbs, the colon absorbs vitamin K, short chain fatty acid, biotin, B5, folic acid, these nutrients that are a result of bacterial fermentation. If we want to talk about the molecular level of the sodium absorption, it's a little bit complex, but you have to know that 90% of absorption of sodium and actively and the passive absorption of water occur in the proximal colon. This is actually regulated by the NAK ATPase channel, and we have the luminal exchange of sodium potassium and chlor bicarb exchange that finally will result in sodium, water, chlor absorption, excretion of bicarb, and potassium. As you see here, this is in the colon, the bicarb intraluminal, and we have here the in the blood we have the sodium. This is under aldosterone, and we have the NAK ATPase. What else? We want in the distal colon, we have what we call it the epithelial sodium channel. It enhances in the distal colon, it enhances the sodium absorption. This is the epithelial sodium channel. Okay, we do not want to go deep. We said these are under aldosterone regulation. Also, I don't want to forget that we have the cystic fibrosis conductase regulator, which is mainly in the distal colon and responsible for the absorption of sodium. And what else? Chloride absorption in the colon, especially in the distal colon, under the regulation of cystic fibrosis conductase regulator. Okay, we don't want to go deep in the molecular level because we will get actually. Okay, this mechanism, this molecular mechanism of absorption of sodium and chloride may explain some diseases such as Clostridium difficile, which can be explained at the molecular level by inhibition of the sodium-potassium exchange. Also, it can inflammatory bowel disease can be explained. Where is it? Inflammatory bowel disease can be explained also by inhibition of the sodium-hydrogen exchange, which ultimately will increase the sodium and chloride inside the lumen of the colon, and water will follow, and so we will have watery diarrhea. And we said inflammatory bowel disease is explained by inhibition of the sodium hydrogen exchange, and also inhibition of the epithelial sodium channel in the distal colon. Also, we have an entity called congenital chloride diarrhea. This is due to a mutation in a gene called downregulated in colonic adenoma, DRA, which will inhibit the chloride OH exchange. Okay, cholera. Cholera actually, its toxin will result in increase in the adenyl cyclase activity. Increasing the adenyl cyclase activity will lead to increase in CAMP. CAMP will actually stimulate secretion, active secretion, of chloride and bicarb, and will result in diarrhea that is high with electrolytes, especially chloride. So you can see here that cholera stimulation of adenyl cyclase increases CAMP. This will increase chloride secretion, not absorption, secretion, and decrease absorption of sodium and chloride. And I think the clue for cholera is the increased intracellular adenyl cyclase activity. Okay, this is just how the malabsorption of sodium chloride may explain some diseases such as infectious diarrhea, Clostridium difficile, inflammatory bowel disease, cholera, etc. Let's move to potassium. We previously explained it. Okay, as we said, potassium is secreted in the epithelium, excreted with the stool. The concentration between the serum potassium level and colonic lumen is the same, 4 to 5 milliequivalent per liter. This is under aldosterone. Aldosterone will increase the secretion of potassium in the stool, and actually not only, we said not only potassium, but also excreted the bicarb, and bicarb may explain why the stool pH is slightly alkaline. And we said that one of the rule of the bicarb that is excreted by the epithelium to the lumen is to neutralize the acidic environment from the colonic bacteria. So, and in cases of prolonged diarrhea, we will have what? We will have hypokalemic metabolic, because we lost potassium, we lost bicarb in exchange with the chloride, so we will have hypokalemic metabolic acidosis. Okay, we said that potassium, sodium, water, all under aldosterone regulation, and it increases the potassium concentration of feces and decreases the sodium concentration. This is by different mechanism in the distal and proximal colon. You can see that sodium concentration in feces can be decreased from 30 to 2 milliequivalent. How does this occur? This occur by the increase in sodium permeability in the brush border, and increase in the sodium pump molecule in the basolateral membrane. Also, it increases the potassium concentration from 75 milliequivalent to 150. How does this effect occur? In the distal, we said in the proximal colon, it is by NaH exchange, and in the distal colon by actively by sodium potassium ATPase. Okay, water, you can imagine that about 2 meters of surface area we have in the colon, and the absorptive surface area is larger. This is because of the crypt. So and we said that it follow the osmotic, hydrostatic also pressure. We have absorption, absorption from lumen to the blood, which is higher than secretion. The epithelium of the colon secrete water, but the absorption is higher, so we have net absorption. Also, water absorption can be regulated under the autonomic nervous system and by adrenergic and anticholinergic stimulation. Okay, short chain fatty acid also we talked about, and short chain fatty acid, these are organic carbohydrate which by bacteria will be converted to short chain fatty acid. It is organic carbohydrate in the colon by the bacteria will be short chain fatty acid. In the proximal colon, butyrate is the most important. It can be an energy source. It's the main energy source for the colonic epithelium. Many effects. One of the main effects that it enhances the absorption of sodium chloride and water in the colon. There is a hypothesis that it can regulate proliferation, differentiation, expression, gene expression, immune function, and colonic wound healing. Also, it reduces inflammation in patients with inflammatory bowel disease and divergent colitis. Hypothesis says that it reduces the risk of colon cancer, and you can see here butyrate. It is primary energy source for the colonocyte. enhance water and salt absorption. Propionate, it participate in gluconeogenesis. Acetate, it participate in production of energy for the muscle. We talked about also vitamin K absorption, which present in diet, but the completion of absorption is in the colon by the bacteria. Actually, it's a lipid soluble. Its absence in the newborn, they have no bacteria in the colon, so bleeding diathesis can be present, and they are given vitamin K. Okay. The function of vitamin K, it play roles in coagulation. It also play a role in signal transduction, cell proliferation, bone and cartilage metabolism. Okay, what else we can add about absorption? Okay, in the small intestine, it can be absorbed, but it is energy dependent mechanism. In the colon, the vitamin K is absorbed passively with passive diffusion. This is two picture just reviewing the function of the colon. We talked about function of the colon: absorb vitamin produced by bacteria. Of course, we said some of vitamin B, not all. For example, vitamin B12 is absorbed in the terminal ileum. Reabsorb water and minerals. Store fecal material prior to defecation. They just review the function of each part. For example, the ascending colon, the transient time when the feces or let's say the chyme, the enteric continent enter the ascending colon, it needs only 2 hours to reach the transverse. This also will give you a hint that the ascending colon is not the primary site for absorption, is not the primary site for storage, it's not the primary site for mixing. The transverse colon, while in the transverse colon, the transient time or the time needed for the feces to transverse the colon is longer, about 1 day, 24 hours. So it is the primary site for removal of water, electrolyte, and storage of feces. Okay. The descending colon, it's a conduit between the transverse and the sigmoid. Rectosigmoid colon, we start to talk about the continence, to maintain the fecal continence. The rectosigmoid colon, actually, physiology, we will highlight it when we will start to talk about the rectoanal physiology. You can see here that the chyme, the transient, when the food enter the cecum up to it leaves the sigmoid colon, it need about 2 days. Okay, this is another picture, shows you that absorption in the large intestine, little absorption actually when we compare it to the small bowel. Most of the absorption occur in the proximal half, and they call it the absorptive colon, while storage occur in the distal half, and they call it the storage colon. The large intestine can absorb up to 5 to 8 liter of fluid and electrolyte each day, especially if there is a absorptive problem in the small bowel. It can accommodate by different mechanism, neurological or non-neurological mechanism. You can see that water absorption, about half to 1.5 liter is absorbed in the colon. What we lose is 100 to 200 milliliter per day with feces. Sodium is also absorbed. We talked about that. Sodium is actively absorbed and potassium is secreted in the colon. Chloride is absorbed in exchange with bicarb. We already talked about that. Vitamins such as vitamin K, biotin, vitamin B5, folic acid, but not vitamin B12. Short chain fatty acid. Some drugs such as steroid and aspirin can be absorbed. Okay, colonic innervation. Okay, colonic innervation. We previously discussed at the beginning of this lecture the colonic innervation, and we said that the nervous system is divided to central and peripheral nervous system, and there is a role of the central nervous system that play in the colonic innervation. And even when we talk about the in the lecture of inflammatory bowel, irritable bowel disease, the hypothesis that exists now that there is a brain gut axis that may explain the pain in irritable bowel syndrome, for example. So this is regarding the central nervous system play a role, but let's concentrate in the peripheral nervous system. Peripheral nervous system, as you see in front of you, is divided to somatic and autonomic. Somatic doesn't play a role in the innervation of the colon, but if we go distal, as we said, in the rectoanal junction, yes, it has role. But in the colon, we are talking about the autonomic. We have sympathetic, we have parasympathetic, and we have the enteric nervous system. We said that the sympathetic is inhibitory effect in the movement, parasympathetic excitatory effect, and enteric, this we already talked about the enteric nervous system, and we said these are the two plexuses, the Meissner plexuses in the submucosa and the Erbach plexuses between the circular and the longitudinal muscle of muscularis propria. And let's return back to the colonic innervation. Okay, colonic innervation. No. This is the sympathetic and parasympathetic. You can see here that the sympathetic innervation of the colon, sympathetic, is from, if we are talking about the proximal half of the colon, it's from the superior mesenteric ganglia. If it is to the left side of the colon or distal half of the colon, it's from the inferior mesenteric ganglion. And the sympathetic and parasympathetic innervation to the distal colon is from pelvic splanchnic plexuses. Parasympathetic, the proximal colon, the vagal nerve, the distal colon, parasympathetic innervation from the pelvic splanchnic plexuses. What else we want to add about the innervation? What is the function of mesenteric plexus and submucosal plexus? Actually, the submucosal plexuses play roles in ion absorption, secretion, regulate they regulate the endocrine and exocrine function of the colon, while the mesenteric plexus, it participates in motility of the colon, in the contraction of the colon. Let's see, submucosal plexuses or Meissner plexus, controlling function within the inner wall. It is in the submucosa. It is responsible for local intestinal secretion, local absorption, contraction in a lesser degree in the submucosal level. While the mesenteric plexus or Erbach plexus, controlling muscle activity along the length of the gut with a specific effect. It increases the muscle tone, increase intensity of the contraction. Actually, one of the differences between the submucosal and the Erbach plexus is that submucosal plexus is composed of excitatory neuron, while the myenteric plexus composed of excitatory and inhibitory neurons. What else we want to add about the innervation of the colon? Let's see, and not to forget anything. This is the colonic innervation. We said sympathetic, parasympathetic, and enteric. Sympathetic to the proximal colon from the superior mesenteric ganglia, distal colon, inferior mesenteric ganglia. If we are talking about the rectum and anus, this is the hypogastric plexus. Parasympathetic proximal colon, it's the vagus nerve. Parasympathetic innervation of the distal colon, we start to talk about the pelvic nerve, and they call it the, we will review them when we will start talk about the rectoanal physiology, nervi irrigant, I think. Okay. Actually, the sympathetic and parasympathetic, finally, this is an afferent and efferent neurons, and we have mediators, excitatory mediators. These are the acetylcholine, tachykinine, and substances B, and inhibitory mediator. These are the nitric oxide, VIP and ATP, vasointestinal peptide and ATP are inhibitory mediator. Okay, what else we want to add? Okay, let's move back. We talked about the Meissner. Okay, let's talk about how does motion and the motility of the colon. Okay, colonic motility. In general, we have two types. In details, it's more complex. Actually, we have two types for colonic motility. These are the segmental contraction or the mixing contraction or the haustration. These occur over a short distance, 2.5 about centimeter, mainly in the proximal colon, triggered mainly by stretch, distension, and it actually increase the absorption. These are due to the contraction of the tinea coli. The propagative contraction, these or the, in another word, the mass movement, the peristalsis, these are responsible for moving the colonic continent toward the rectum and anus, occur over a long distance, about 20 centimeter. Many trigger for this movement: stretch, one of them; irritation; reflexes such as gastrocolic reflexes also play a role. They can divide, they may be divided, the propagative contraction, to low and high amplitude propagated contraction. High amplitude propagated contraction is the one that is responsible for moving the colonic content toward the anus. It increases after meal. What else we can add? Before defecation also it increases. We have a concept called the colonic tone. The colonic tone is due to the actin and myosin interaction. Also, it is under neurological control, and the tone may increase and decrease depending on many factors. Of them, the content of the or the composition of the colonic content. For example, high carbohydrate and fat content will increase the tone, while the protein and amino acid will decrease the tone of the colon. Okay, what else we have? We have another concept about the motion or the motility of the colon. It is the burst contraction. Where is it? This is it. The contractile burst. It is actually a sudden movement of the colon. In another word, it's called the colonic motor complex. It's not clear what's the function of this contraction. It's present in the colon and also in the rectum, rectal motor complex. These are not clear and can occur 1 to 2 per hour and last for 6 minutes. What I think that we have to remember that we have two main type of colonic contraction, and let's move and discuss briefly modulators of colonic motility. Okay, I don't want to disturb you with the modulator of colonic motility, but actually it is the gate to many pharmacological, therapeutical treatment for patient who suffers from diarrhea or constipation, especially in patient with irritable bowel syndrome. So, and even we discuss many of this information in the lecture of irritable bowel syndrome. Of these stimulatory neurotransmitters are the acetylcholine and substances B, and inhibitory neurotransmitters such as vasointestinal peptide and nitric oxide. And as you see, we previously discussed this agent, especially when we talk about the inflammatory bowel disease. These can treat constipation dominant irritable bowel syndrome or diarrhea predominant irritable bowel syndrome. And here we have the sacral stimulation, which is proven for a treatment of fecal incontinence. Okay, microbiome, the last part of this lecture, and we actually talked about the importance of the microbiome and the reflection of the function and physiology of the colon. We said that it may participate in synthesis of many vitamins, short chain fatty acid, etc. So we will talk about microbiome. And microbiome, it means the bacteria that present in the colon, and it's large amount, about 110 trillion in the intestine. They present in a healthy patient in the lumen and in the mucosa, in the epithelial surfaces. The composition of these bacteria and the variety of this bacteria depends on the diet, lifestyle, usage of antibiotic. Of course, we have a commensalistics relationship. We depend on them in certain function, and bacteria also cannot live without us. Of this function, as we will see, they participate in production of short chain fatty acid and vitamin K, some vitamin B, synthesis and absorption of amino acid. They start to appear period after birth, and this may explain the bleeding tendency in newborn. Also, as we said, composition depends on diet, lifestyle, also on the acidity and the O2 along the GI tract. Diversity is an index of a healthy patient. This is not 100% correct, because some healthy food can decrease the diversity of the bacteria present. There are proven that microbiome changes, which we call it dysbiosis, with certain diseases such as irritable bowel disease, inflammatory bowel disease, colorectal cancer, autism. We do not know is these changes are causative of this disease or a consequence, effect of these disease. Of the microbiome, 98% of them are bacteroides and firmicutes. Inflammatory bowel disease, we have a lecture, we talked about the microbiome and the studies regarding the microbiome, regarding the inflammatory bowel diseases, and this may lead us to some treatment with antibiotic, prebiotic or probiotic to change and to alter the microbiome in the bowel. This may benefit or not. It's not 100% clear. The study is developing regarding that, but what we know, for example, that in Crohn's disease the diversity of microbiome is lower than healthy patient. We have increased in Crohn's disease, increased bacteroides and proteobacteria, while in ulcerative colitis we have proven that there is a decrease in Faecolobacterium prausnitzii. Okay, what else we want to add regarding the microbiome is the pronunciation was good about Faecolobacterium prausnitzii, which is, as we said, decreased in or associated with patient with ulcerative colitis, absence in patient with ulcerative colitis. Okay, this is a nice picture, shows you the function of the gut flora and highlights some other functions also. For example, synthesis of vitamin K and some group of vitamin B. We said vitamin B12 is absorbed and in the proximal bowel. Folic acid, biotin, thiamine. B12, it's not synthesized there, maybe. Okay. What else? Here, the bacterium. Okay. Formed vitamin K is important. Okay. What the other? The second function, deconjugation and decarboxylation of bile salt. Third, breakdown of bile pigment to produce stercapilinogen, which is responsible for the color of the stool. Decarboxylation of amino acid to produce amine and histamine. The amine are excreted in feces and responsible for the smell. Breakdown of urea by bacteria urease to ammonia. Actually, the bacterial urease produced by the bacteria will actually convert urea to ammonia. Ammonia will go to the liver, and if there is liver failure, there will be built up and hepatic encephalopathy can develop. Fermentation of undigested oligosaccharide. Some complex oligosaccharide will be fermented, and you can see here that colonic bacilli are capable for ingestion of small amount of cellulose. This is a nice information. Ammonia is normally converted to urea. Ammonia converted to urea in the liver and cleared out of the body through the urine. In individual with acute or chronic liver failure, there will be a build of the ammonia, and this will lead to hepatic encephalopathy. This is regarding the microbiome of the colon. If you need more information, we discuss it in the inflammatory bowel disease. We highlight in the importance of the microbiome. We reach the end. We said that it participates in short chain fatty acid. Yeah, let's check that we do not miss any information. Yeah, okay, thank you. We reach the end of our lecture.